You're listening to Tatiana is Everyone, an Orphan Black podcast. My name is Chris.
And my name is Stephanie, and this episode is going to contain some spoilers for the series through all 5 seasons. Just so you know, if you haven't seen the show in its entirety, this episode could contain spoilers for the entire series.
And in this episode, we are interviewing the authors of The Science of Orphan Black, the official companion, Casey Griffin and Nina Nesseth.
Yes, we were really excited to get to talk with them. We read the book and we both really enjoyed it. There's a chapter on cloning, on chimerism, on Rachel's brain injury and recovery, as well as a chapter on transhumanism, which is the idea that the human race can evolve beyond its current mental and physical limitations like we see in the Neolution movement. The book covers all of the big topics as well as answers common questions that Clone Club has had over the seasons, such as should Katya's fingerprints have pulled up that match for Sarah's? Spoiler, no, and Casey and Nina explain why in the book. And we felt very privileged that they were willing to talk with us about the book and Orphan Black and all sorts of great science-y things.
Yes, all true.
Would y'all mind introducing yourselves?
Sure. My name is Nina Nesseth. I am a professional science communicator. I'm based up in Sudbury, Ontario, Canada. And my background's in biomedical biology, but of course I'm a huge fan of Orphan Black.
And I am Casey Griffin. I am getting my PhD in developmental and stem cell biology at the University of Southern California in Los Angeles. And my science background is basically in genetics, developmental biology, and stem cells. And I'm also a huge fan of Orphan Black.
So how did y'all come to be writing this book, talking about the science of Orphan Black? How did you become attached to this project?
Oh gosh, so this is actually, it's kind of a funny story and very true to how Clone Club sort of works as a community, because Casey and I met at the first San Diego fan meetup that BBC America held for an Orphan Black fan meetup. And Casey had won an Orphan Black trivia contest at the event. And my friend and I had won a Allison Hendricks theater scene recreation contest where we had to kind of act out a little skit from one of the scenes. And so Casey and I got to go on stage together and meet the cast and take a bunch of photos and get some Orphan Black swag, which was, probably amazing. It felt like I sort of peaked, right? In terms of fan experience. And we didn't talk at all.
We were too overwhelmed.
Yeah, I think we were in a group hug together, but we did not talk at all. And then the internet Clone Club kind of realized that we should be friends and connected us over Tumblr. Casey, for a while, she'd been writing these Orphan Black Science Time Tumblr posts that I had admired from afar. A little bit later on, I had gotten the opportunity to pitch the idea of writing Orphan Black science recaps, for every episode for the Mary Sue. And I couldn't do it without Casey, so we did that together. And that was pretty much the launching point for our book. We ended up getting approached by our agent, Maria Vicenti from PS Literary Agency. And she was like, hey, your recaps are awesome. This needs to be a book. From then
Maria
on,
is awesome.
It was amazing. It was one of those things where we had to take a step back and be like, whoa, we can write the book.
It was like a crazy idea we hadn't even considered.
But we knew that if we were going to do it, it would be the both of us writing it together. That was about 2 years from when we decided to write the book. It was about 2 years, I think, that we were working on it.
Yeah, it was right after season 3 had aired. So yeah, about 2 years.
Yeah, and then it got picked up by ECW Press, and they were able to make connections with Graham Manson, and it became the official companion. So it wasn't always the official companion for the show, but it became the official companion with Graham and with Cosima Herder's blessing.
Awesome. That's like a fandom fairy tale or something.
Right?
Much.
And so I was very impressed that you were able to incorporate so much of the stuff from season 5 in a book that came out pretty quickly after season 5. So did y'all sort of have a sense of what was going on in season 5 a little bit before the rest of us did?
Yes, we were told we were told the general science ideas a while ago because Cosima Herder had at least known that they would be focusing on like longevity and life expansion. So we had a general idea, and then once they finished writing the scripts for season 5, they sent them our way so we could include all the necessary information, which was pretty awesome.
Yeah, I think we had the general idea around like September, so before they started shooting. But we were getting the scripts around January, February, I think, and it was brutal to have to know everything and not tell anybody. Yes. Except each other. And I would read faster than Casey, so I had to like read it and then sit there and be like, Casey, have you read it yet? Have you read it yet?
I was stressing her out so much. I'd be like, oh, I'm too busy. I only read one. She was like, no.
And then she'd text me, just like capital A's. I knew she had read what I had read.
This sounds like Chris and I.
It does.
Chris usually got to watch the episodes before we did. She's like, I want to talk to you, but I can't.
Yeah. A lot of our conversations, even when we're watching the show, a lot of our conversations back and forth, because I would read the scripts first, but when it came to actually watching the show, Casey would always watch it first. So a lot of it would just be like a lot of screaming at each other over text messages.
Incoherent, babble, nonsense texts. It was, that's how we communicate.
Spoil each other, but we got all the spoilers.
So something I really appreciated reading the book was I felt like it gave me a much better sense of the big picture of kind of the science storylines of the show. Because I feel like most of the time I understood the individual revelations or beats as they were playing out. But then as I was reading your book, I realized, I hadn't made that connection of this reveal in season 5 to this connection to this reveal in season 1. So thank you for that, for really painting a nice big picture of the science of the show.
Yay.
Thanks.
That's awesome. Yeah.
I was actually wondering what topics that you covered in the book were you most excited to talk about?
Oh gosh.
For me, it's always I just love talking about Cosima. So the clone disease in and of itself was exciting for me. Also, personally, the stuff about creating the cure and all of that is the closest to the kind of work I do. So I looked forward to writing about that just because it felt like writing about what I do every day.
Yeah, all the genetic stuff is really Casey's bread and butter. I think probably the chapter I was most excited about was kind of going into the neuroscience elements of like Rachel's injury and recovery, because I'm all about brains. I'm all about anatomy and neuroanatomy. But like surprisingly, something that surprised me was how much I loved going into the history of a lot of the science in the show, because Orphan Black did a really good job of kind of sneaking all these nods in everywhere, some more obvious than others, to major experiments and history that kind of led to our understanding of science today. And also like making sort of comments on the sorts of problems and biases that history has introduced into our understanding of science today. And it's, I was surprised in how interested I was in digging into sort of like those scientists and science philosophers that are like big names, but you don't really pay attention to outside of your textbooks.
That actually kind of leads me into another question. I was wondering if there were things that they brought up in the show that you had to research to see if they were true or how true they were. Because, like, the eyelid bone growth thing
horrifying.
Is horrifying, but also comes to mind in this category. Because I feel like I heard something about that, but maybe never knew if it was actually a thing.
Because it doesn't sound really real. You're very likely to dismiss it when you see it on the show. But so I was very curious about your case study feature on Crystal, where you talked about that.
Right. It sounds totally like some sort of like magazine that you see at like the corner store that's just like Lady grows
Alien.
bones in her eyelids. They click like castanets. That example was one that we kind of had to look into a little bit more, but it was built on something where I was like, I have heard of this case before and read about this case and about those sorts of crystals kind of forming and bone formation kind of forming in response to the different chemical components of different cosmetic procedures. But I think the one that I had to do the most digging into outside of the clone disease, because the clone disease it's a novel disease that they created for the show, but it's based on a lot of different diseases in certain ways. So a lot of that was kind of like piecing together the different elements from different illnesses.
Like a puzzle. Yeah, it was a puzzle to figure out what they took from where and how it all worked together.
Right.
It's fun.
But I think it was probably, in terms of like how much of this is real, how much of this is based on something real, was the Neolution maggot bots. Especially the maggot bots is like a gene delivery, like a gene therapy delivery system, because it's like a horribly inefficient way to to go about that sort of treatment. Yeah, that was an interesting one, trying to like dig into like different like entomology textbooks. I have a colleague who's an entomologist, and I just held up a picture of the maggot bot to him, and I was like, "Hey, Dan, what kind of maggot is this?"
Did he know?
I learned this. The main thing I learned was that maggots are really hard to identify because they all just kind of look like maggots, unless you can like get right in there and like count cilia, like count little hairs on them and stuff.
Wow.
Yeah. So now you know something more about maggots.
Fantastic.
All our first conversations about the maggot bots had to do with like real life organisms that will be that sort of parasitic entity that also might deliver some sort of genetic material. It was all looking into all sorts of weird bugs. Ugh.
Yeah, like there's I remember I found that one study that looked at schistosomes, like a type of worm that they were using.
Those worms, yeah.
Yeah, they were like using their eggs or using them in general to look at treating different, I think it was bowel conditions.
Mm-hmm.
And they were proposing them as a potential treatment because they're parasites and they're really good at hiding from the immune system. And I was like, But like maggots aren't exactly like, they don't have that same sort of skill set, right? Like maggots not going to put, great at hiding from immune system on its resume. But
I mean, it might.
We all lie on our resumes, don't we?
Well, right. Yeah.
These ones are altered already. They're cyborg maggots.
It's disgusting.
It sounds like, Casey, you did not enjoy your etymology class very much.
I'm not a big bug person. It's kind of gross.
Well, I think they're cool.
That was like my worst nightmare, was that scene when he
Dr. Nealon
spits the worm at Delphine. Yeah, ugh.
I was going to say, season 4 was not kind to you then.
Yeah.
Yuck.
I'm glad you brought up the maggot bots, though. That was actually one of my favorite sections of the book, particularly because you have a little section where you talk about why this sort of maggot bot model kind of makes sense, talking about like the parasitoids versus parasites and the fact that it might be able to hide from immune systems and things like that. That was really fascinating to me. Gross, but fascinating.
Yeah. I mean, like right now, we do have probably better vectors, but like it's so neat to think about like how this could work if someone decided that was, the way to go for a gene therapy delivery system that could like live inside a body.
Yeah, you mentioned that it was an odd choice to put it in the cheek, since that's a fairly vulnerable place for it to be. The only advantage seemed to be that it was close to the brain. Did you have any thoughts of where it might be better placed in the body?
That's a great question.
I mean, I would think something even just like your arm or your leg or something, because your cheek is so, there's so much wear and tear in that area. How many times do you bite your cheek accidentally while you're eating? And so that just seems like such a delicate place.
Yeah, even just like somewhere more internal, like I don't know. I was guessing somewhere more in your like abdominal cavity or something. Just
yeah,
right,
right into your stomach or something.
Just like tuck it right into like your mesentery or something like that. Just like just give it a little like cozy place in between like the folds of your intestines or something, somewhere like that's more internal where you're less likely to, damage it by some sort of impact or by any sort of like mechanical injury. Because like you're especially since like your torso and your abdomen are kind of like built to protect squishy things. It just seems like a better idea to tuck it in somewhere in there where your body's already kind of like built to protect it.
It's true, and also it occurs to me it sounds like Stephanie is planning on implanting maggot bots, but doing it better than Nia Luchin did.
I appreciate efficiency, is all I'm saying.
Yeah, just find the good artery, and so that if it's delivering through. Oh, because you have to have that like neurotoxin fail safe too. So you want it to get pumped around quickly. Hook it up to some artery, and then tuck it somewhere where it's not going to get chewed on by accident.
This makes perfect sense to me.
Also, it looks like Stephanie is making notes mentally. I'm a little worried.
Another thing that really sort of struck me at the end of the book was I kind of felt fuzzy about Project Castor, about the purpose and what was going on there and stuff like that. I felt that way already. I feel like reading your book, I feel that way even more because I feel like you introduced a lot of questions about, they kind of hinted at what they're doing over here, but they didn't really say, and they might have been doing this, but they didn't really say. So do you similarly feel a little confused about Project Castor?
Yeah. So Project Castor, as far as it's sort of elucidated in the scripts and in the show, was like we know it was an offshoot of, well, not quite the same project as Project Leda, but like Cody was kind of running with it. Eventually, it's like that's the thing. It's like it's unclear whether from the beginning she was planning to exploit that Castor pathogen as this sort of eugenics tool, we'll say. But like we know that eventually that's what it became, if not what she was planning from the beginning. But it was just like it's sort of unclear as to how she was going to implement it, if they were just going to like, just send the casters out into the world, or if they're going to, turn it into some sort of kind of side-by-side project with what we saw with the Blue Zone cosmetics, where it's like delivering gene therapy through cosmetics, or are we also creating like a cheaper variety that's delivering a pathogen through cosmetics? Like,
I think some of the confusion comes from the fact that I think the clone disease kind of disrupted all of that work, and whatever the goals were initially, it became a big problem that this disease was killing off the clones, and so Cody had to switch all of her efforts towards that. And that's the point that we saw. So we didn't really get a good view into what the purpose exactly specifically was before they ran into these issues.
Because we know, like, from the get-go for Project Leda, like, we know from Ethan's side, he was more on the side of just saying, like, oh, we're creating clones as proof of concept just to see that we can. And then we know from Susan Duncan's side that she and Westmoreland were introducing synthetic sequences in hopes of kind of manipulating the Lin28A gene to have longer lifespan, and seeing if that manipulated gene would manifest, if the traits would manifest in that clone generation. But then, like, what Cody was running with, they never say that they're trying to do like the same sort of experiment with the Castor clones. And since she's kind of like run it as military, it sounds like they're kind of trying to weaponize something from the get-go. We don't know if there's a different sequence that they introduced and we're experimenting with the Castors, because nothing like that's ever said. And basically, especially in season 5, as soon as they sort of established that, Lin28A is the focus, they kind of said, okay, well, we found it. We know that's manifested in the surprise daughter generation in Kira and in Helena's babies. And so Leda's one, we're just going to completely ignore the Castor experiment and then just terminate it completely, which is really interesting, because you would think that they'd still have some sort of ends with Castor. If their goal was using that pathogen, you would think that they would still be useful in some way. But basically, their story just amounted to the idea that the Castor clones were just too glitchy to be successful, and so they just terminated the experiment.
I don't know about you guys, but I always got the feeling watching the show that the scientists were just kind of like, well, since we've got this going, why don't we just do this too? Like I know that sounds weird. The social aspect to Project Leda, because it is so
messy.
Yes, it's messy and really random and not that carefully orchestrated. It just feels like somebody in the group was kind of like, hey, can I do this too since we've already got this running?
Yeah.
and that's kind of what I'm getting
you
with
mean
Castor.
like the whole fact that they sent them out into the world and had monitors to kind of observe
Right.
their development? Okay, yeah.
Well,
no,
'cause I think you mentioned at some point in there too that it seems really... It's not good science, because like the monitors there's no set pattern for the monitors, so
the monitors can do whatever they want.
Yeah. Y'all sounded super skeptical about the social aspect of the Leda experiment when I was reading that. Like they sound pretty skeptical of how this was all set up. But I was talking about it with my spouse, who's an anthropologist and a qualitative researcher, and even she was like, yeah, I mean, they're not trained, the people they have monitoring the clones. You don't know how they're going to collect the data, what type of data they're going to collect. It's just not a well set up experiment.
Yeah, like,
no, you're right.
Why would you do that? Because, yeah, if the point is to make an experiment, you need as few variables as possible. And they're just like, we'll have a spouse over here and a secret lover over here and whatever.
And just, like, this one's going to know what's going on, but not this one.
But this one's not, And this one knows everything, and this one's kind of sketchy, but hey, we'll use her anyway.
Yeah, it's like just such a great way to skew yourself.
I mean, it did seem like they had a very thorough worksheet.
They did have a very thorough worksheet. You do make a good point, Chris.
Yeah.
They just, I don't know. It was kind of like, whatever.
Like, who thought it was a good idea to make Donnie a monitor? Like,
no. But do you think there could be usefulness to that data that they did collect, even though it was very, the collection itself was not particularly well executed?
I mean, they had a lot of observation of the different clones, so they were able to, like, really distinguish the differences between them. But a lot of those differences could have been, like, observed in other ways, too. Speaking from, like, the more traits aspect of it.
I guess personality-wise.
But even then your reporting is really biased.
Right. But I mean, at least in the case of Allison, like, well, you would think that her spouse, if anyone else, would know her best. Obviously, he kind of didn't, but, you would think.
Yeah, and then there was, like, the usefulness of, like, sending, like, the lab teams in the middle of the night. But that's a collection that could have been done differently.
That was the only thing I could really think of, is that perhaps by having the monitors, it gave them more direct access to taking samples from the clones, potentially. Because I suppose they could have gone through doctors if they were trying to keep them naive to the clone experiment. But I don't know. That was the only thing I could come up with.
Just needed somebody to unlock the door.
Because it's really cool to think about, like, the amount of data you could get from doing a longitudinal study with clones. Because, like, we have a few famous, like, longitudinal studies, like twin studies, like the Minnesota twin study that we talk about in the book, that has given us a lot of insight, if not, like, concrete information, at least a lot of insight into, like, kind of what shapes a person who is pretty much, like, genetically identical or close to it at birth. And to have clones, it's, like, almost as good as it can get for really seeing how nurture and other elements influence a person. But it's just such a messy experiment, even with everything kind of, like, really controlled and contained, just with, like, so many variables that, like, it's already a really tough thing to measure and to report on. And the fact that they kind of just threw whatever kind of Joe Blow monitor at some of the clones to do the reporting just makes it even messier. So I'm really curious to see what Neolution is doing or has done with all that information. Like, they have all these files, of, like, almost 300 clones sitting in, drawer somewhere or in the file somewhere at Dyad, and, like, what are they using it for? What have they used it for?
So an aspect of the book that I really liked was the different case studies where you used one of the clones to talk about a particular topic. And some of them, I feel like they felt rather like a logical thing to talk about. Like with Tony, you talk about androgen replacement therapy. With Helena, you talked about like thought reform. But I was wondering if you had any other topics that you maybe toyed talking about in connection to the clones. Because like, for example, with Beth, you talked about like SSRIs and SNRIs. Makes sense to talk about that in regards to Beth, but maybe there were some other things I thought you might have considered talking about too. So I was just wondering if there were any like discarded case study topics.
We definitely did. Some of them were, like, as you said, Tony was just like obvious that we should talk about androgen replacement therapy. But for some of them, I remember with MK, we definitely toyed around a couple ideas. We thought about talking about maybe autism because it was revealed in the comics that she is autistic. And there were a couple others. With Crystal, we definitely threw around a couple ideas. Allison was at first a bit difficult, I feel like, as well.
Yeah, we had a hard time finding what to talk about for Allison.
Beth, I kind of felt like we always wanted to talk about some aspect of depression because, you know, it's a very prominent part of the show when she jumps in front of the train. So it kind of made sense to talk about something in connection with that for her. But yeah, it was definitely, I feel like MK, Crystal, and Allison were definitely the ones we talked about the most.
And I know we had another topic for Cosima that I was
right.
lobbying for, and I can't remember what it was. Because you were like, no, I've got this. And I was like, but what about this for Cosima? And I can't for the life of me remember what it was.
Yeah, whatever it was, you would come up with the idea and you told me about it, but I had already like done some of the work for the whole like medical marijuana stuff. So I was like, oh, I got this. This is fine. But yeah, I can't remember what it was.
Whatever it was, I was really excited for it.
Yeah.
For the Sarah case study, you talked about Sarah as the wild type, which is a comment that Cosima makes on the show. And I hadn't really made the connection to the fruit flies, because I'm like, oh, that's why in biology class, when we're doing little cross thingies, we use capital W's and lowercase w's when we're talking about the flies, because it's referring to the wild type of the little fruit flies. And something that you mentioned in that section about, in the case study of Sarah, was the idea that they might have left Sarah and Helena fertile on purpose to sort of stand in as a wild type to compare to the mutations as the clones. I hadn't really thought about that as an option before.
So that scene where Cosima says that she's the wild type is my favorite scene in the whole show. I'm like such a dork for those two and their like sister relationship. I just can't even handle it. So that was actually one of the Tumblr Science Time posts I wrote, just talking about Sarah as the wild type, not as much in detail as we go into in the book, but it definitely was something I always thought about, because for every experiment I set up in the lab, you always have to have a control that is as close to whatever would be considered wild type. It just felt like it made sense that there would be a clone that was more wild type than the rest of them. And ideally that would be someone with like no synthetic sequences, but I guess even just making them more with the ability to be fertile and just like different from the rest would be, you know, something you would think they would have thought of. So, yeah.
Mm-hmm. Clearly they haven't been left alone with all of their normal or like, you know, original genome, because they, you know, both Helena and Sarah have, you know, Kira and Donald and Arthur, who have, you know, that manipulated gene. But like in terms of how in their generation their traits are expressed, like they're both fertile. So yeah, no, it's just a cool thought experiment. And it would make a lot of sense, as Casey said.
I had a question about Kendall and the chimera thing.
Love it.
The chimera thing. They spend a whole chapter on the chimera thing. It's kind of complicated, but yes.
Well, it's just the fact that she's a chimera. They make a big thing of it, But can you explain to me if that is a factor in her being chosen as the
original for the
original source? Because they make it sound like it is, but if it is, I don't quite understand how.
Okay, to be honest, I'm pretty sure, and Casey, you can say whether you think I'm right or not, but I'm pretty sure it's just like when they were going through, looking at prison inmates and taking swabs of them, and when they discovered Kendall's chimerism, I feel like with Ethan especially, they were just like, that's so cool. We got to use her because her biology is so cool. And
that definitely seemed like what it was to me. Yeah. It's like they didn't go into it with the plan of finding a chimera. And I feel like, they probably went into it with the idea that they wanted to have this like XX clone line versus an XY clone line. But who knows? Maybe they were just in it for like one clone line, and then they come across Kendall and they're like, well, this is awesome. Like, let's just run with this.
Yeah, I totally think that's what it is. Because outside of the chimerism, there's nothing about Kendall's cells or her cell lines individually that are especially special.
Yeah. They talk about how they had to do the manipulations to like Lin-28A for them and like all the other synthetic sequences. So it seems like, Kendall was just nothing special except that she had 2 different cell lines inside of her.
Yeah, like she wasn't like Giannis with a cool gene already.
Yeah.
Or gene variant.
So this is perhaps me just speculating out of a lack of scientific knowledge. But you do
speculating because you don't know any better.
But you do mention you go through like the sort of the interesting idea of the chimeras of a chimera's immune system, because it allows these 2 cell lines to exist in one body without attacking the foreign cell line. And do you think, is it possible? Are people asking, could that potentially make a chimera more suitable for this type of cloning experiment? Because there's like manipulation of putting parts of one cell into another with like somatic cell nuclear transfer.
It's definitely possible because, it's a very unique situation because it's different than like conjoined twins because they are identical twins, so it's identical cell lines, so there's no immune system problem. This is really like a person with another piece of some other person inside of them, and that doesn't get attacked. Most of the time a chimera doesn't even know they're a chimera until something happens where they accidentally find out about it. And so it definitely would, a person who's a chimera would definitely be an interesting subject to look at when you're thinking about doing these sort of manipulations. They would definitely be an interesting person to do, maggot bot studies on.
I'll make a note.
Add that to your notes.
Casey, actually, because I don't know the answer to this, but do you know if there is any sort of increased risk or any risk at all for immune reactions with SCNT? Because it's just like a single cell being manipulated, right? It's not...
Right. It's a single cell, so there's none of that. There's no specialized, immune cells or anything. But there is still... there is still like a fail rate because it just won't always work. When you try to take a nucleus from another cell and put it into this cell, it'll sometimes just be like, no thanks. So there's definitely going to be a fail rate. But so I don't know if a chimera cells would be any different than anyone else's for simple SCNT.
Yeah, because my suspicion is that it wouldn't really, because it's like separate from the body's immune system and that body's immune function, it probably wouldn't have an effect.
That's kind of what I figured, but I thought I'd ask the question anyway.
No, it's a great question.
I am using my editorial privileges here to clean up a couple of questions that I asked extremely poorly. I was nervous and rambly, and I didn't write down my questions with enough details. I want to say thank you to Casey, who did her best to answer my terrible question before I could clarify it. So I had a question about the ID sequences included in the clones' genomes, such as Cosima's 324B21. You know, in Biology 101, we learned that one of DNA's big jobs is coding for proteins, but the ID sequences are non-coding. I asked Casey if she could explain how non-coding sequences work. And then I wondered if the dyad scientist worried that the ID sequences could create differences in the clones, given that each of the ID sequences would be unique.
Only about 3% of your genome codes for coding sequences. So only about 3% of your genome will be turned into proteins. And that requires what's called a promoter, which is a region before the gene that is kind of the part of the DNA that preps the gene to be transcribed. And then it also requires the transcription start site, which is just a codon, a methionine that's like, hey, this is where you start. And the protein, the polymerase, will bind there and start the transcription. All the rest of the DNA that's out there is, at least most of the rest, is regulating that process, whether it's repressing it, enhancing it, changing it in some way. I mean, these could be sequences that are extremely far away or even on different chromosomes that control a gene, but they'll come into contact, the chromosome and the DNA will fold and bend to make interactions. So a synthetic sequence can be made to code for parts that'll be marked for activation or repression. And then if you put that into the genome, these activating or repressing molecules will bind there and then cause them to have these sort of functions. You could totally change a person's transcriptome, like the way genes are expressed in them, just by changing non-coding sequences. Although the clone disease prion, that is what attacks the uterus in the Leda clones and the brain in the Castor clones, is a coding sequence because they needed to make this completely new protein. I think the ID tag sequence in itself is a relatively small sequence, and that's one of the sequences they had a specific location that they knew they wanted to put it. They wanted to attach it to cytochrome C. So they would have made a way to insert it into the genome where it would insert either right before or right after this sequence. And so when the cell's machinery went to transcribe cytochrome C, the ID tag wouldn't get transcribed. It would be after a stop codon or before a start codon. It wouldn't be in the promoter or any other regulatory regions. They would probably just insert it after the end of the genomic sequence, so it wouldn't be touched by the cell's machinery.
And we see real examples of that, like currently, even now, like you see stories all the time of using DNA as data storage, like, oh, we put like all of Shakespeare's works inside this sequence. And so it's not out there to do that. A little bit different inside a human being, but yeah.
And I mean, they do it with all the like transgenic animals that are used in laboratories. They all have these sequences in there that under certain situations won't disrupt the biology. A lot of the manipulations people will do to study specific genes and specific developmental systems are set up in a way so they can have a control where this gene isn't disrupted, and then you add a drug to the animal and then the gene is just disrupted. So unless a certain element like a drug or a certain protein or whatever is there, then there's nothing. It doesn't get transcribed. It's not affecting the biology in any way.
It's kind of like the biological version of when game developers would like hide their credits in the middle of a game.
So kind of like putting it in little comment quotes in like HTML. It's there, but it doesn't get shown in the web page or whatever.
Right. Yeah. You can find it if you're looking for it, but no one will know it's there if they're just checking it out.
Just scrolling through. Yeah.
Yeah.
going to veer fandomward after the science talk for a moment. Because you mentioned transgenic, and I noticed that there was mention of Dark Angel in the book. Who's the Dark Angel fan?
Me, Nina. I remember, well, I watched it when it first aired, but also I remember when I was in high school, it was in syndication and reruns were showing, and I had like a fourth period spare, and I just like walk home during my spare and watch Dark Angel and then walk back to school to like go work on the yearbook committee. Like, so yeah, that was definitely me.
I was just wondering, because I also am a Dark Angel fan, and I feel like I should get Stephanie to watch some of it, and we can do an episode about it. Because
yeah, I haven't seen it.
I think it would be interesting.
Oh, it's totally camp. But yeah, you should check it out.
Oh, it's pretty ridiculous, but thematically there are similarities, I feel like.
Absolutely, yeah. And some similar questions that get asked too about like the good old like ethics of playing God with people and manipulating their genomes and that sort of idea.
Mm-hmm. And ownership over self and that sort of thing.
Yeah, absolutely.
So Nina, you had mentioned that writing like the Rachel chapter where you talk about the effects of her brain injury was one of your favorite things to write. I also really appreciated that chapter. The thing that caught my attention first, though, when I was looking at like the list of the chapter titles and things, was that it was called "My Poor, Poor Rachel," is a quote from her father, Ethan Duncan. And I kind of thought, ooh, interesting, because I feel like Rachel's a very polarizing character for a lot of people. Some people think she can be redeemed. Some people just think she's totally awful. So I thought it was interesting that your chapter was titled that, and I was curious to see like what tack you were going to take talking about her. Do you have like strong feelings about her just as a character aside from like the science portion of her storyline?
I love her as a character.
Yeah, me too. I love Rachel. We often, like Casey and I, when we're talking about different clones, we often say that like Rachel's one of the most interesting clones to think about.
For sure.
And like really think about like who she is as a person and how she fits into all of this and her motivations, and like just as a character, she's fascinating. And in terms of like, I don't know, I'm not one of those like people who are in the boat of like, you know, Rachel just needs a hug, ooh woo, like like that's not where I am with her. I think that
she enjoys being who she is. I feel like she would never. It's not just a like, oh, everybody abused Rachel. Like she makes choices.
A lot of her choices were like born of her circumstances, but like to a certain extent too, you have to ask like a lot of these traits that she has and how like angry and manipulative, not to mention how like manipulated she has been. It's just like asking like how much of that is born of her circumstances and of her environment, and how much of it is just who she is as a person. One of the most fascinating scenes actually is in season 5 when Rachel and Allison finally face off, and I was so glad for Allison's remark that we're not so different when upon Rachel saying that she wanted to put her hands around Allison's throat, because like, yeah, I've been arguing for a while that like if Allison had been brought up in Rachel's conditions, they probably would be very similar people.
Allison and Rachel are so similar in like personality wise. I feel like it's just they were brought up completely differently, so it manifested completely differently.
Like I almost wish there had been another clone raised self-aware somewhere else, like in an isolation of Crystal, and not Crystal, oh my god, of Rachel. Imagine if Crystal had been raised self-aware.
She still wouldn't know it. They would have told her she was a clone.
She was a no.
Yeah, they don't look like me at all. What are you talking about?
No.
Like that is the longitudinal study I want to see, is like two self-aware clones raised in isolation of each other and in like naive of each other, but otherwise
they don't know there's another one.
Yeah. Where's that experiment, Dyad?
I think you make a good point, though, because I think we see in both Allison and Rachel, even though they were raised in sort of different circumstances, they both have that same tendency to not express their anger or try not to express their anger until it becomes just so much they can't help it and they kind of explode to some extent in often very potentially self-destructive ways. Like when Rachel smashes the stem cells that they'd gotten from Kira. And I thought it was I hadn't really thought about it in this regard, because I think it's easy, well, at least in Rachel's case, to be kind of like, well, she's awful, of course she would kind of do this. But you talked, when talking about her brain injury, how potentially her actions at the end of season 4 toward her mother, when she stabs her mother and then stabs Sarah, could have been effects of the brain injury to her frontal lobe.
Yeah, like that, there's so many ways with brain injury where, how it can impact people and impact their functions. But just the fact that it was pointedly in, like her frontal lobe and like her orbital frontal lobe can kind of, like there is a precedent for cases of, much reduced impulse control. And with someone like Rachel, who already has greatly reduced impulse control in periods of like, high emotion, it's not that far out there. But I think what we also mentioned too is like one of the more common effects or impacts of brain injury is it's usually depression in some form. Mm-hmm. And absolutely, we saw that in Rachel. Like Rachel's already the loneliest clone, but in most cases, that's what you're going to see, in part because of the injury and in part because of, a person's capacity being reduced in some way from an injury and having to deal with that and learn how to deal with that. And the work that you have to put into it and the emotional workload of having to work with that change in your person.
Think about it now. There is that scene early in season 2 where we see her have sort of a flash of just wrecking her office, which I wonder now if they were planning on doing this when they wrote that scene, sort of as an indicator of the things that she wants to do but isn't.
Yeah. Like, these are internal now, but they're going to be external potentially in the future.
Mm-hmm.
Yeah.
That makes me think of, like, and the fact that that's not what she presents to people. That's actually another cool parallel between Allison and Rachel, is that they both want they both want the gold star and the pat on the head. And they don't really care so much about, like, Allison a bit more than Rachel, but they don't, their motivation isn't to be good people.
It's to seem like good people.
But, like, yeah, it's about, like, their motivation is for people to think, to see them as what they want people to see them as. Like in Rachel's case, seeing her as this powerful person in this position of power. She wants that veneer and will hold on to it as much as she can. And Allison's the same way. She wants to be seen as the model citizen, you know what I mean? And she will cling to that with like every last fingernail.
Yeah, they both are very approval seeking.
Oh yeah.
So do you have a favorite clone, Nina? Casey has expressed quite a bit of affection for Cosima. But
yes, always.
So do you have a favorite one?
Well, it's a hard question to answer. So yeah, like Rachel's my favorite clone to think about. Narrative-wise, Beth is probably my favorite for narrative, just because I loved that for the first, up until season 4, we only knew Beth as either what people said about her, or through like Sarah's imagined version of Beth, and through like people's like dreamscapes. Like we never actually saw who Beth was as a person. She was kind of just this like this being created out of other people's ideas. So I'm just like in love with Beth as a character in that way. But I have this like really, really special affection for Allison. I love her so much. I love her. I cosplay Allison at cons, and
like perfectly though.
Have you been Allison at Dragon Con in the past few years?
No, I've never been to Dragon Con. I would love to. No, I've been Allison at mostly San Diego Comic-Con.
I did see the pictures. You're very good.
Oh, thank you. Thanks. Yeah, and at Fan Expo in Toronto, and New York Comic-Con I did once, and I think that's it.
I was just curious, because we have seen a couple of Allison cosplayers at Dragon Con, so maybe it was Nina. Anyway. Oh, that's so awesome. I love seeing other cosplayers, especially Orphan Black cosplayers. I saw just a few weekends ago at Fan Expo. Oh, last weekend. There were a lot of really great cosplayers, and there was like Cosima in a tux with her Delphine. Nice.
And they were great. So good.
So do you think you're going to stay with more traditional Allison look in future cosplay, or are you going to adapt to like her season 5 more
the purple hair?
Oh my god.
Freewheeling appearance.
The stage mom haircut.
Carry around a keyboard.
Yeah. Her funky fresh new purple dew.
Liver deep.
You know what? A lot of people have asked me that, and I don't know. Like right now I'm a staunch no, because my Allison is the Allison who, is hyper anxious and critical of everyone and walking really fast and covering up for other people's mistakes and her own mistakes. And not so much, new chill kombucha drinking Allison. Although I will say that I do love the moment that she has when she first takes out the keyboard, the Korg, and she like turns on a preset and is like bobbing her head like it's like the coolest, like most inspired jam ever. So I might cosplay it just to like steal someone's keyboard and just like recreate that moment. But I don't know.
I feel like she's most recognizable, which is sometimes hard when you're doing like characters that look pretty much, like anybody else on the street. I feel like she's more recognizable as sort of season 1 through 4 Allison, but maybe you could do like one day of hippie freewheeling Allison and the rest of the days of the con you're like regular Allison.
Oh, people never recognize me when I cosplay Allison unless they're like diehard Clone Clubbers.
Yeah.
Because I just look like this person who accidentally wandered out of J. Crew and into the convention.
Who's that soccer mom?
Yeah, like, what are you doing here? Usually I have to like stand next to my Casima. Like I have a few friends who do a group cosplay with us, and like, yeah, if I'm next to any other clone, it doesn't work. But if I'm standing next to Casima, people always recognize Casima, and they'll be like, "Oh my gosh, Casima!" And then they'll ask for a photo with her, and then ask like their bag. And I'll be like, everybody loves Casima. And they'll be like, oh, whoa. To be fair, my friend does a killer Casima. Like, she's great.
Yeah, I feel like Orphan Black cosplay does tend to work better in groups. Helena's pretty recognizable by herself, especially if you're wearing the wedding dress.
I was going to say, we did at Dragon Con see a wedding dress Helena.
It was really good.
She was terrifying. It was amazing.
Good.
But let us know if you need a liver deep temporary tattoo, because
we can hook you up.
We have a friend.
Please? That's awesome. I would love that. It's really funny. The one time I cosplayed Helena once, and it was more as a dare. Like I put together a Helena cosplay in like 6 hours, and it was really fun. But I cosplayed MK once, and that was probably the funniest one because it got the most reactions, but people didn't know who she was. So a lot of people shouted "The Purge" at me.
Okay.
I feel like I would be unnerved if that happened to me.
Yeah.
Yeah, it was really alarming. And this was at San Diego Comic-Con, and I was in one panel where Felicia Day was hosting, and I got to ask a question, and she didn't know what I was from either, but she called me Animal Head. So like it was interesting. And I know like the real MK would probably have had like her heart like swelled 3 sizes to have Felicia Day talk to her in any way. Yeah, it's just really funny doing like clone play and not being recognized at all.
So, Casey, do you have any other clone favorites besides Casima?
Casima. But okay, aside from Casima, I love Helena. I absolutely love her. I loved her from the moment we met her. If Casima wasn't there, I would be totally obsessed with Helena because she's just everything. She's amazing. Like she is to me, the best mother like you can imagine, but she's also gonna like kill that drug dealer, no problem. And it's just like perfect. I love her.
I concur.
Yeah. And I would just love to sit down and eat an entire donut store with her. That would be pretty fun.
Yes, Chris refers to Helena as her precious murder angel.
Because she's my precious murder angel.
That definitely works.
Also, I spend too much time on Tumblr, apparently.
Yeah. Did you have any other questions you wanted to
I don't think so.
Okay. I feel like there was one that was tickling the back of my brain, but now I don't remember what it was. Okay.
Was it a question about brains?
It was not a question about brains. Though I did, speaking of brains, when you were talking about brains in your book, I did kind of go, oh. When you're talking about
was it the
leaving...
your brain is really mushy passage? Because that was one of my favorites.
Yes, like your brain, you can see the impressions of your fingers in the brain after you hold it. That was the part.
Just because of the weight of the brain itself.
Yeah.
That was a good descriptor.
Yes.
Yeah. There are videos of like people holding fresh brains on YouTube if you ever want to check it out.
Some late night.
You see just how like soft it is. But yeah, please, like that's why helmets exist. Your brains are really squishy.
I feel like that's a good place to end. Wear a helmet, folks.
It's true. Wear a helmet.
If we've learned anything from Orphan Black, it's wear a helmet.
Even if it's a backwards motorcycle helmet.
I was going to say, do they have to be fur trimmed? Because that might be tricky for me. I'm not good with a glue gun. Allison, can you help me?
Nina has a glue gun.
Yeah, I have 2 glue guns, so I can help you out.
Thank you.
You're welcome.
Well, thank you both so much for speaking with us. Are there any things that anything you'd like to plug on the podcast before we conclude?
I mean, we can like shamelessly plug our book, but
well, where can people find the book? Do you have any other projects that you're working on as well that are Orphan Black related potentially?
Not currently, maybe sometime in the future. Right now, we're focusing on our lives a little bit. I'm getting married next weekend and just bought a house.
Congratulations.
Congratulations.
Oh, thank you. And and Casey's finishing up her PhD.
Yeah. Woo, fun.
But yeah, like the Science of Orphan Black is out now, which is super exciting. And I think it's also having release in more countries this month in September. You can find it, on Amazon or
BarnesandNoble.com.
Yeah, through your local bookstore, or you can find it on our publisher's website, which is ecwpress.com.
And we will link to that in our show notes.
Or you can call my mom and ask her to give you one of the 10 copies she bought. So yeah, my mom's got some extras.
You know what everybody in your family is getting for Christmas.
Yes, exactly.
Well, thank you both so much for taking the time to speak with us today. We really appreciate it.
We really do. Thank you.
Thank you.
Thank you.
Just to talk for a minute about our other podcasts, our final Wynonna Earp Season 2 discussion is live on our multi-fandom podcast, Finalysis. Our friend Annie and Stephanie and I discuss the final 3 episodes of Season 2. You can listen to it now and find out how to subscribe at askgenretv.com/fan.
Also on our Killjoys podcast, Season 3 of Killjoys has just wrapped up recently. You can listen to our episode discussions of Season 3 and find out how to subscribe over on our website, askgenretv.com/Killjoys. The name of that podcast is The Quad, also, if you want to find it on your favorite podcast app.
We don't always say it that way.
The Quad, you mean?
The Quad.
I like saying it that way.
I know you do, but we don't always say it like that. We would love to hear your thoughts about this interview, or if you've read the book, your thoughts on the book. You can send them to us, feedback@tatianaiseveryone.com. We love getting voice messages, which you can send us in a couple of ways. You can record a voice memo on your smartphone and email it to us, or you can call our listener voicemail line at 972-514-7223. You can follow us on Twitter at TIE Podcast, and we are also on Tumblr and Facebook.
Tatiana is Everyone is part of the Ask Genre TV family of podcasts. To find our other podcasts about Wynonna Earp and Killjoys and some other shows, visit our website, askgenretv.com. And in this episode, the abdominal cavity was played by Tatiana Maslany.
I'm so sorry, Tatiana Maslany. Thank you for listening.